Hello everybody, my name's Sam Sheppard and it's a pleasure to be here today, and I'm going to be talking about some work that I've been doing on to try and understand how agricultural intensification relates to the spread of pathogenic Campylobacter. So work in my group focuses on a range of bacteria actually, and really what we're interested in is understanding the ecology and evolution of pathogens. So as you all know, I'm sure many of the most famous bacterial pathogens actually exist principally as commensal organisms. So in my group we will typically use genomic techniques to understand the changes that occur as pathogenic forms emerge from this background of commensal ancestors. And here are just some example organisms that we worked on. Obviously Campylobacter, but also you can see Staphylococci, famous for MRSA and among other diseases and infections. Bacillus cereus in the middle, E. coli on the bottom left, lots of famous E. coli diseases, and Helicobacter pylori at the bottom right, which of course has strong links with dyspepsia and gastric cancer, etc. Okay, so today, of course, we're talking about Campylobacter. You all know, or I'm sure that many people here today know as much about this organism as I do, and many know a lot more, I'm sure. But obviously, it's very common. It's among the world's most common bacterial pathogens, gastroenteritis pathogens, especially, of course, Campylobacter jejuni and Campylobacter coli. And you may or may not know that actually infection caused by these two organisms is 3 times more common in high-income countries, for example, the UK, than infection caused by E. coli, Salmonella and Listeria combined, which is kind of strange because they tend to be a little bit more famous organisms. But anyway, and obviously there's a big, significant cost associated with campylobacteriosis. But of course, you will also know, I'm sure, that usually we don't think of Campylobacter as transmitted from human to human. So this is where thinking about the potential source comes in as a way of trying to basically to reduce disease. So Campylobacter is principally a commensal organism of warm-blooded animals. We've sampled it, myself and many other people who are in this meeting, sampled it from all different sources. Typically, you know, where we look, we tend to find this organism. So to reduce transmission to humans, we might want to ask, okay, well, how does Campylobacter evolve to survive in these lots of different hosts? And also, obviously, another big major challenge is where is antimicrobial resistance acquired? Because, of course, although most human infection is self-limiting, some severe infections require the use of antibiotics, and Campylobacter, just like many other organisms, is gradually getting more and more resistant to the various drugs that we use against it. So when we're thinking about Campylobacter in its natural habitat, in these warm-blooded animals, you know, you can think, you know, what we need to think and remember is that not all animals are equal in terms of niche size. And these are quite startling statistics. If you look on the left, we've got a picture of a chicken and an owl. What we're saying here is that actually, if we estimate the biomass of all birds on Earth, actually 70% of all bird biomass is in poultry. Okay, and actually for me, even more sort of depressing, I suppose, is if we think about mammalian biomass on Earth. Obviously there are lots of humans, but 60% of all mammalian biomass is in livestock. So this is a lot of cows, but also sheep, goats and pigs. And only 4% of mammalian biomass is in wild mammals. So, big deal. What about the impact? Well, if we just think about cattle, okay, then certainly Campylobacter are the most abundant bacterial pathogens in cattle. They inhabit the gut, and they're present at around 20% of cattle, although that's completely an estimate. And they're found at a concentration of around 3 times 10 to the 4 colony-forming units per gram. Okay, these are just estimates, but this is just an interesting way of thinking about how many Campylobacter there are on Earth. There are around 1.5 billion cattle on Earth, each of which produces about 30 kilograms of manure every day. Okay, so we can do a little calculation. Of course, there's massive error one way or another in this, but we can estimate that there are around 3 times 10 to the 17, that's 300 quadrillion Campylobacter excreted into the environment every day. Okay, so just the sheer magnitude of this shedding leads to lots and lots of environmental contamination, and of course the risk of contamination of human food production chains. So myself and lots of other people on this call, in this meeting, have worked a lot on this over the years, trying to understand the relative contribution of different reservoir hosts to human infection. The simple principle really is that by looking at multilocus sequence typing genes, or now more commonly whole genomes, we can identify genetic variation that is associated with a given host, a given reservoir host. Then, if we see that variation in a clinical isolate that's come from an infected human, then we can make a prediction that it came from a particular reservoir host. Okay, very simple principle, but of course lots of work has gone into this, and we've used it to create models where we can actually assign a probability to a given host source. I'm sure many of you know all about this, and the take-home message, okay, certainly from the work that I was lucky enough to do with Ali and Martin and Fran when I was in Oxford, really showed that chicken was and Noel, of course, sorry, showed that chicken was a major source of human infection. It's certainly in the UK. Possibly there's recent work, possibly there's a bit more infection from cattle in France, maybe, but chicken, big source. But little really is known about how intensive farming influences the evolution of these organisms within their host. Okay, and that's really what I'm focusing on today. So if we think about a Campylobacter tree, this is just a tree made from different Campylobacter strains, and the tip of the tree is labeled by the host source. So it's yellow, that isolate came from chicken, where it's blue, it came from cattle. Well, if we look at this tree, we can see clusters of strains that are quite closely related that are found in cattle and chickens, okay? And we can think of those as ecological generalists. But in this recent work, what we showed is that some of the ecological specialist strains, okay, so these are Campylobacter that found only or mostly in only one host. So one of those, the cattle-associated ST61, seems to have emerged from this background of generalist strains. And, you know, by looking at the genomes of lots and lots of isolates, we were also able to identify the adaptations that potentially allow those strains to exploit that cattle niche better and give them a competitive advantage in that niche. And we're able to show that these are acquired through horizontal gene transfer, and there's gene gain and gene loss and all kinds of variations that help a Campylobacter survive in cattle. And these might be related to diet or host physiology or factors associated with habitation, like high stocking densities. Maybe a strain that can survive, maybe strains don't need to be able to survive outside of the host to transmit terribly well. We've done work on that as well. And in this slide, I'm just reminding us, while chicken is a major source, an estimated 500,000 annual cases of human Campylobacteriosis are caused by the two most common cattle-associated Campylobacters every year. But this is the bit that I sort of really wanted to talk about today, and I'm running out of time already, but it's good to have these short, punchy talks, in my opinion. So when did these host specialist strains evolve? That's one thing that's very interesting to me. So did they evolve in antiquity when cattle were first domesticated, 10,000 BC, or is it more recent? if you look at this graph, we can see an estimate of the number of cattle on Earth from a long time ago. Again, of course, this is quite a crude estimate, but obviously the number of cattle on Earth has increased dramatically in recent years. So when did host specialism evolve? Well, if we zoom in on the right-hand side of the graph, which is what we've done here, and we sort of expanded it, and we've overlaid, so the blue dots are still the number of cattle on Earth, but we've overlaid a tree of this famous ST61 cattle-associated Campylobacter lineage. And what you can see, actually, is that this lineage didn't emerge until, well, we estimate, you know, sort of the 1800s, and the massive majority of diversification in this lineage has occurred in the last 30 years or so. Okay, and I think perhaps an image of me is sadly over the top of the right hand side of the graph, but anyway, it goes up to 2010. So this sort of tells us that agricultural intensification has had a major impact and actually coincides with the emergence of these specialist lineages. But what about the rise of antimicrobial resistance? I mean, it's not just me, but this is just an example of some work that I did. But the rise certainly in ciprofloxacin, so fluoroquinolone resistance in animals coincides with resistance in humans. Okay, and this is kind of strange on one level because we already said Campylobacter is self-limiting, and we don't think of humans as human-to-human transmission as common. So humans are a dead-end host. So where is the selection for resistance taking place? If it's not within us, then, you know, where is it taking place? Well, we know that there's lots of use of antibiotics in agriculture. Perhaps the practice of using antibiotics as growth promoters, well, certainly it's not very common now in high-income countries, I suppose. But it's still quite common in some countries. And then, of course, there are sick animals as well that need antibiotics. And there are global transmission networks. So there's a possibility that antimicrobial resistance could evolve within agricultural animals and then be passed. So they pass the resistant strains to humans. But really proving this is quite difficult. So I just wanted to mention briefly some work that we've done where we've characterized the antimicrobial resistance genes in Campylobacter from animals, sewage, and humans. And if you just look at the bottom circle here for aminoglycoside resistance genes, if you look at the sort of red curve at about 10 o'clock on the outer circle, what that is lots of different aminoglycoside resistance genes. And where there's a red line emanating that to another different colored curve around the outside of the circle, that means that exactly the same gene and exactly the same allele was found in a Campylobacter strain from another source. So you can see just by looking at that example that the same aminoglycoside associated resistance gene was found in Campylobacter from humans and C. jejuni from humans and cattle and chickens, but also the same gene was found in C. coli from chickens and humans. So this doesn't tell us definitively that where antimicrobial resistance evolves, but it certainly provides us with clues about the transmission of resistant strains, so clonal transmission, but also the transmission or horizontal gene transfer of resistant genes. So that would be gene pool transmission. Okay, so I've run out of time. I've gone over a little bit. I hope you'll forgive me. Just to summarize, today I've given a sort of whistle-stop tour of ways that we're trying to understand how agricultural intensification is influencing Campylobacter spread. We've talked about the numbers game that leads to much more spillover, but also some of the more nuanced ways in which agricultural intensification might be influencing adaptation. Is this the final story? Of course it's not. It's not enough here for us to think about it might help us to think about targeted interventions that might reduce transmission to humans, but in my opinion, we need a lot more information on source sink dynamics, and especially information about the people who are at the greatest risk of Campylobacteriosis, which of course are people in low and middle income countries where chronic diarrhea is a massive problem, especially for children, causing growth faltering and morbidity and sometimes death. Okay, so thank you very much, and I think I should be able to take some questions.